Good afternoon, everybody. theraCUES welcomes all participants to this webinar, an exciting webinar on miRNA. So miRNA molecule has been the focus or in focus for the entire month. You must have seen a lot of posts on our LinkedIn also. Today we have Dr. Vinitha Richard with us. Welcome, Dr. Vinitha. And I would like to introduce her to all the audience. Dr. Vinitha Richard earned her doctoral degree in biotechnology from Rajiv Gandhi Centre for Biotechnology in Kerala, RGCB, and with the research focus on tumor cells, stem cells regulated by microRNA in oral cancer. Post PhD, she received her first grant as an ICMR DHR woman scientist to work on tumor stem cell fractions in metastatic breast cancer. She relocated to Ireland to work as a senior postdoc in Precision Oncology Ireland (POI) Consortium under the mentorship of Professor Michael Carrin at Lambay Institute for Translational Research, University of Galway, Ireland. Her current research goals are to identify molecular biomarkers that are precisely track tumor epithelial cell transitions for the early diagnosis and prediction of prognosis in breast cancer. Welcome, Dr. Richard. I am your host for the day, Vijay Lakshmi Bhatt. I also head the lab at theraCUES. And over to Dr. Richard.
Thank you so much for the kind introduction. So I think without much delay, I would go to share my slide. So hopefully you can see the whole screen.
We are able to see your screen, Dr. Richard.
Yeah, thank you so much. So getting on, as you can see, the title is microRNAs as mediators of cell state transitions in breast cancer. And reflecting on the title, I couldn't help but notice like transitions happen everywhere, every time. It's like a day-to-day occurrence. So a major transition that happened in my research career was when I moved to Ireland. So as you can see, I'm currently based at the Lamb Institute for Translational Research over here. And exactly behind this building, you can see the University Hospital Galway. So this center is in close vicinity to three major landmarks. That is the University of Galway, formerly known as NUIG. And this is the architectural quadrangle, which is quite famous, and the Galway Cathedral. Now, our proximity to hospital actually supports our research in cancer. Now, moving towards my focus on cancer, why is it so significant? So the significance relies on the incidence rate. And as you can see, breast cancer ranks as the most predominant organ or the site of cancer in female. And relatively, the mortality rate is also quite high. Now, the paradox of breast cancer is that though it is a disease that affects a simple gland, and we have made major advancements in therapy, screening approaches, diagnostics, but still the high incidence shows that we definitely need to take a different approach. Now, though we know the molecular subtypes and has been clearly defined histologically, we face the hurdle of sudden emergence of different subtypes based on the gain or loss of receptors. Now, this can also be attributed to the role of heterogeneity happening both within the tumor as well as between the patients. And now to top it off, we do have heterogeneity occurring at the molecular level. Now, I will be just walking you through each of these aspects shortly. Now, as I mentioned early on, sorry, the slides are running away. As I mentioned early on, the breast organ as such is a simple organ made of luminal epithelial cells surrounded by a myoepithelial lining and protected from the stroma by a basement membrane. But when you look at the tissue, sorry, the slides are, okay, I hope it's okay now. I think a timer is set here. Okay. So when you look at the tissue hierarchy of a normal mammary epithelial cells, it has been noted that there are different grades of progenitors that that progress towards differentiation. And at each stage, each of these cell types start expressing different keratins as well as hormone receptors like vitamin D receptor, androgen receptor, ER receptor. When you look at the overall constitution of the tissue, it shows the presence of almost 10 to 11 luminal epithelial cell type. So you could definitely imagine, like, when a cancer hits at this type of a tissue hierarchy, what all components that can emerge out of it, the subtype combinations that can come out of this type of a tissue hierarchy. So keeping this complexity in mind, we are progressing further and introducing the breast tumor heterogeneity as such, as I mentioned previously. More than the interpatient heterogeneity, you do have an intra-tumor heterogeneity, which is accounted based on the interaction with immune cells, the interaction with the epigenetic landscape, the genomic instability, as well as the clonal selection that happens with therapeutic regimen. So to proceed, there is one major disadvantage that actually is disadvantageous to us, but is advantageous to a breast cancer cell. That's called as the hormone receptor discordance. Now, as you can see, the blue circle is basically the percentage of occurrence in primary tumors, and the yellow slice is basically the ratio of metastatic samples. Now, if you look at the hormone receptor positive status like ER, PR, and HER2, HER2 has less percentage of mets, but the gain of receptor is quite high in HER2 when compared to PR and ER. Now, another striking factor that you can notice from here is that the PR status has a higher metastatic potential, so indicating there is a connectivity with the breast cancer initiating cells and this hormonal status. Now, in case of ER and PR, majority is the loss of receptor. And to correlate this heterogeneity with hormone receptor discordance and drug resistance, it was noted that most tumors actually encompass both ER and PR clonal cells. Now, as the patient is subjected to neoadjuvant endocrine therapy or chemotherapy, what happens is that post-surgery, there is an emergence of drug-resistant clones, which might have lost its receptor expression completely, becoming a new subtype. Now, this can lead to micromets as well as macromets in later stage. Now, the main attribute to this is post-translational or transcriptional regulation of the mRNA expression through microRNAs. And as you all know, they are like small 19 to 25 nucleotide long single RNA threads that can actually bind to any messenger RNA target and block the production of protein. So this eventually changes the phenotype of cancer cell. Now, the lab where I'm currently based has a long-standing history where they noted that microRNA could predict the receptor status of breast tissue. It could be utilized to detect early-stage breast cancer from blood, and they were able to determine the tumor subtype based on the expression in tissue and circulation. And also, it could predict the responders from the non-responders to neoadjuvant chemotherapy. With this background knowledge, we were doubtful, like, is this restricted to only the hormone receptor positive subtype, the microRNA-based regulation? But we were surprised to see that even HER2 receptor, which is quite targetable, it's still indomitable because of the major role it plays in a dual regulatory mechanism with microRNA. Now, as you can see here, there is a microRNA encoded very adjacent to the HER2 gene, MIR4728, which itself can go and block the signaling pathway from estrogen receptor. Now, the HER2 gene itself, the HER2 receptor itself triggers signaling pathways, which trigger the production of more microRNAs that can also play a major role in determining the signaling cascade that leads to disease progression in breast cancer. So in turn, almost all the receptors or hormones do regulate microRNAs, and microRNAs in turn undertake a feedback regulatory mechanisms to determine the subtype of breast cancer. So looking at the limitations of currently if we focus on microRNAs alone, we miss out on crucial information pertaining to the intra-tumor heterogeneity, the cell of origin of tumors, and the emergence or the rapid emergence of resistant fractions. So what we propose is look at it in the bottoms-up approach, where you can determine the different cell types first, understand the phenotype, the functionalities, and then proceed to find out what exactly are the molecular regulators that play a role in addressing this heterogeneity, thus trace it back to the cell of origin, and also be utilized to identify or pinpoint the exact emergence of a resistant fraction. Now, the presence of multi-state transitions was not new. So even from my initial research career stage, we proposed this cancer stem cell shift hypothesis way back in 2010, wherein we did an extensive literature review, and we found that at a given time point in a dysplastic or a hyperplastic tumor, this was proposed with oral cancer as a model, where we found there could be an existence or a coexistence of 3 types of cells, like a chemosensitive, highly proliferative stem-like cells, chemo-resistant, highly proliferative stem-like cells, and chemo-resistant dormant stem-like cells. Now, why we called all of them as stem-like cells? Because in a hypoplastic condition, when we try to do a phenotypic staining, almost all the cancer cells were positive for the stemness-associated markers. But that doesn't mean almost all of them are all stem cells. So the presence of this chemo-resistant property was attributed to the expression of drug transporter proteins. So with that in mind, we went ahead performing some experiments with oral squamous cell carcinoma as the model, and we published this paper in 2013, wherein, if you could see, we deployed a technique called site population assay. That is, we stain the cells with Hooke's dye. And I would like to acknowledge Professor Margaret Goodall over here, because she was the pioneer who standardized this technique in bone marrow cells. And she was able to screen hematopoietic stem cells just using just this technique. Now, as you can see, the gate and flow cytometric sorting panel were marked as P3, or the lower gate, is the site population cell that used to reflect the enriched for stem cells. Now, in our study with solid epithelial cancer cells, we found that there are major fractions. So until then, almost all the studies were gating the rest of the fraction as a main population or a non-site population, and they were performing all the experiments. So we found that this particular fraction was similarly enriched with drug transporters, and they were like another version of site population cell. So we called it as SP2. And they contained a higher DNA content than the site population, main site population, or SP1. Correspondingly, there was a non-drug transporter expressing fraction. We called it as main population, or MP1, and the corresponding to SP2, we called it as MP2. Now, we performed functional assays for all these fractions, and I'm not going into too much detail of all the experiments because it's done on oral cancer model. So what we did was that, we went ahead and performed in vivo tumorigenic assay in non-skid mice, and we also did functional characterization. And as you can see, each of these fractions could repopulate the other fraction when cultured for a few days. So, but when we injected it into non-skid mice, only the MP1 fraction did not create any tumor, so it was non-tumorigenic, whereas the other fractions had relatively larger tumors as it progressed towards the MP2 fraction. Now, when you look at the phenotype, each of them were positive for CD147. The SP2 and SP1 were positive for ABCG2, whereas the MP fractions were negative for ABCG2. But the most important point was that they had a different aggressive behavior and the morphology as well. From SP2 to MP2, there was like a transition towards the EMT, and the cells were more mesenchyme-like. The SP1 and the MP1 retained the epithelial morphology. So we understood that there could be some signaling network that is happening at the molecular level. So to understand that, we did a mRNA and miRNA array for the comparing the tumorigenic versus the non-tumorigenic cell. And then we found that there definitely is an interaction or a distinct pattern that predicted like the difference between these fractions or the progression between these cell states. So this is like a snapshot of just the microRNAs and their corresponding mRNA targets derived from the array done on SP fraction, SP1. And as you can see, there is a significant number of downregulated microRNAs in SP1, whereas there's a large number of microRNAs that was upregulated significantly, and their corresponding mRNA targets. Now, as you can see, the pathways were common, but the targets were different for each of these scenarios. And there was, if you could see, there is CD151, which is quite upregulated because the microRNAs that target were downregulated in SP1. Even you can see CD44 over here. And the upregulated expression of a panel could change the expression of CD44 eventually. Now, the presence of multiple cell states in solid epithelial tumors was not only found by us, but it was also reported around the same time by other groups. Now, the irony is that many of them focused on signaling pathways, but not to microRNA-based regulation. For example, the presence of HER2-positive circulating tumor cells in the blood of breast cancer patients. So what they found was that when the cells or the patient is subjected to oxidative stress or was subjected to chemotherapy, it led to the enrichment of a HER2 negative CTC, which survived this treatment and later caused remission. A similar phenomena was found as proliferative heterogeneity in AKT low cells. So in a normal condition, in a cancer patient, the AKT level was high as a proliferative counterpart. And when it was subjected to combination therapy, it led to upregulation of the AKT low cancer cells, which survived those treatment. So this cell state transition was predominant, as well as it was like a sudden transition happening in the midst of the treatment regimen. So it causes a major concern, as we have to halt the therapeutic regimen and then opt for another in the midst of that clinical decision-making scenario. Now, even another publication that was reported in 2013, this was like the CTC in breast cancer patient showed both phenotype as an epithelial cell as well as a mesenchymal phenotype. And when in response to treatment, they found that the mesenchymal CTCs actually suppressed the mesenchymal marker expression and started behaving like more like an epithelial CTC. And as the tumor progressed towards mets later on, they emerged back as a mesenchymal phenotype. So this transition has been happening at different levels in the body in a cancer cell, but we have not been able to address it accurately so that we could target the cells accordingly. So we thought of bringing in the concept of microRNAs, hormone receptors, the stem cell concept in breast cancer. Now, why we were moving towards breast cancer is that we found that, as you see, like the incidence of breast cancer was quite high, and the majority of breast cancer are luminal subtype. And even though we have targeted therapies or endocrinal therapies, 30% of patients fail to respond to this treatment regimen. And the main reasons that they attribute to this therapeutic failure is receptor discordance, as I mentioned earlier, or probably due to the presence of resistant stem-like cells. Now, estrogen as a hormone does not have a direct role in guiding stemness, but it was found that progesterone receptor do play a role in expanding the cancer stem cell population. It was also reported like microRNA-mediated ER-alpha expression can lead to an acquired resistance to first line of endocrine therapy. So based on this, we set up our experiment and our objective. And the first objective was to identify, are there stem-like cells in a hormone receptor positive breast cancer cell? and are they capable of undergoing phenotypic transition? Now, we were also interested in looking at the molecular regulatory mechanisms mediated through microRNAs, which can lead to the sudden emergence of receptor variants. Now, the experimental setup was pretty straightforward, as we had done this previously with the oral cancer. We were focusing on whole breast tumor tissue, but due to some technical issues, we changed to utilizing established breast cancer cell lines. And then we did immunophenotyping across using cancer stem cell associated markers. We did a site population assay on the fraction. We were able to sort out the fractions based on the SP1 or MP1, based on the differential expression of drug transporters. And these fractions were injected in non-skid mice. And the xenografts that were generated was later used to do a histopathological analysis. We did H&E staining and IHC staining for hormone receptors. And based on that expression profile, we categorized the samples and performed a microRNA profiling on the xenograft tissue. And we did bioinformatic analysis and target prediction of the same. Moving on to the results. The first step was we did an immunofluorescence staining for stemness markers, as I mentioned, 147 and 151. We used 5 different cell lines, which represented the known subtypes of breast cancer. And as you can see, more than 80, 85 to 99% of cells were all positive for the stemness markers. So as I mentioned, not all these markers are stem cells. So we did a staining for CD338 is ABCG2 and CD243 is P-glycoprotein or MDR1, which are drug transporter proteins. And you could see like MCF7 and SKBR3 had a differential expression of these drug transporter proteins. So we went ahead and did the aldehyde dehydrogenase assay to screen for the presence of stemness enriched fractions in them. And to our surprise, SKBR3 and MCF7 both had 5% of the cells that were ALDH positive, indicating the percentage of stem-like cells in that group. So when you look at the performance of site population assay, we performed it with MCF7 since it's a ER positive cell line. And as you can see, we can see the hummingbird structure with the beak forming the site population fraction SP1. And there was a fraction of cells which was sorted further away from each other, like the MP1, MP2, and this is the SP2. So these fractions were injected into non-skid mice, and we retrieved the xenografts at 85 days, 90 days, and 120 days post-injection. At the initial step, we found tumors to be generated in all samples, and the unsorted control was used as a control to ensure like there is no inhibition occurring by transferring the cells through high pressure flow cytometric machine. Now, when we took these tumors at 120 days, what we found was that the SP1 tumor was so small, like we could not utilize it for any experiment. And as you can see, the SP2 was relatively small in comparison to the one with MP2 and MP1. So this shows like there is a high proliferation in MP2, and as the days progress, the behavioral pattern of these cells started to take a distinct approach. Now looking at the, H&E staining of these xenograft tumors, you can see like we have the unsorted control, which is more hyalinized. And we kept a normal mouse mammary tissue to compare the size difference between MCF7 and the mouse, so we can rule out contamination with mouse tissue. Now, as you can see, SP1 is more like a poorly differentiated one, whereas SP2 and MP2 fractions had more duct or lobule-like structures. It was more like a well-differentiated tumor. And MP1 had like a partial EMT with Vimentin-like cells. There was evidence of angiogenesis in blood cells penetrating into the tissue. We performed immunohistochemistry staining for ER, PR, and HER2. And this was done on, now I'm just putting up a representative panel where it is all positive. So we did comparison between the technical replicates of xenografts. So we had grouped it. Group 2 was the unsorted control at 85 days and 120 days. And as you can see from the parental cell line, we could see like an emergence of PR negative fraction. Now, the ones highlighted in yellow were quite significant because it caught our attention. Now, the SP1 fraction tissue, the tumor tissue extracted at 85 days. the sp2 xenograft extracted at 120 days, and the mp2 at 90 days, it suddenly turned to a triple negative phenotype with a loss of ER and PR. So we were not sure, like, is it happening only at the protein level, or is it happening at the gene level, or are the mRNA or gene expression not happening in this particular cohort? So to ensure that, we performed a real-time PCR analysis for the ESR1 related genes, and we did a binomial logistic regression of the average of gene score to predict the presence of ER gene, and we put a cutoff of 0.68. Now, what we could see was that almost all the fractions we extracted from xenograft tumors all had a ER score of 0.9. So this was way above, and it was all uniform, indicating that ESR gene expression is definitely there. The transcripts are there in the nucleus or the cytoplasm, but we do not see the expression of protein happening in the tissue. And we also did an aggression analysis to see if these cells are aggressive or not. And to our surprise, almost all the fractions really came higher than the cutoff score of 0.24. So we did a microRNA analysis to see, does microRNA play a role in determining this difference in gene expression? And we performed encounter human miRNA panel with 799 mirs. And as you can see, the SP1 and SP2 were clustered together, and the red indicates the basal phenotype. Surprisingly, the MP2 microRNA profile was more similar to the MCF7 parental control. So this panel itself is predictive, like there is a distinct microRNA pattern emerging in the various fractions arising from the MCF7 cell line. So this shows clearly the molecular heterogeneity occurring within a seemingly homogeneous single cell line. Now, we just compared between the ER negative fraction and the ER positive parental fraction. We did a bioinformatics profiling to see how this particular microRNA could control ER pathway. And we found that this particular mir 29B3P was quite upregulated in our ER negative cohort in comparison to the ER positive cohort. Now, what we understood was that it was reported to be quite upregulated in CD44 positive breast cancer stem cells, and it was also reported to be a promoter for the development of the triple negative subtype. And an inhibition of this particular microRNA actually prevented migration or the metastatic potential of MDMB231, which is a known metastatic cell line. So what we conclude was this particular microRNA to be upregulated in our ER negative subtype fraction indicates a transformation of cells to one that is enriched with malignant stem-like cells. To summarize, this was what we proposed and we published in IGMS in 2023, that from one single cohort of cells, if we consider this as a patient sample, we can extract different combination of cell lines which are like chemo-resistant dormant, chemo-resistant hypoproliferative, chemo-sensitive hypoproliferative, or something which is similar to the bulk of the tumor that can succumb to the drug treatment. Now, each of these fractions had distinct phenotypic profile, enlarged nuclei, the angiogenic potential, invasiveness, and the proliferation potential. So at a specific time point, our cancer stem cell hypothesis really stands because there is a mix of different subtypes in one seemingly homogeneous cell or a cancer or tumor tissue. To summarize, we would like to say that a whole profiling of microRNA across tumor tissue or in circulation definitely can support, gives a global picture that can be utilized for detection, but subclassification of cells or the distinctive features that you can categorize based on their hormone receptors or the stemness potential, that will enable us to address heterogeneity and design targeted therapy in such a way where we have a complete remission of the disease. Now, as I mentioned early on, the part of this work was done in RGCB and then was done in back-end lab institute. So I would like to thank my former mentors, Professor Radhakrishna Pillai and Dr. T.R. Santosh, for supporting the study over there. My former colleague, Dr. Sarah Jones, and Dr. Vishnu, who was the veterinarian over there. He is currently pursuing his PhD in breast cancer. And this work would not have been completed without the support and the collaborative effort of Dr. Jyoti and Dr. Madhu, specifically Madhu from St. John's Research Institute, and the industry partner, Dr. Gopal Krishna, and his theraCUES team, who actually provided support with the miRNA array and the bioinformatics analysis. So last but not the least, I would wish to thank my current mentor, Dr. Michael Kirin, and also the patients and donors here in Galway who are really kind-hearted and supporting our research, and for NBCRI for funding support. And I was part of the Precision Oncology Ireland project, and now we are moving on to phase 2 of it. I also wish to thank the hosting center, that's the University of Galway, and the Cancer Biobank, the faculty over there, the discipline of surgery team, and I have a research assistant, Kelsey. So we form a good team over there, and we hope our research becomes more fruitful as the day goes. So thank you so much. And this is the Lamb Institute when you see it in December, like around 6 p.m. So we are now moving towards the cold climate over there. Thanks again, and I'm happy to take questions.
We are open to questions for Dr. Richard.
Yeah, I can see a few chats coming in. Aggressiveness cutoff was set. I'm sorry, such low as ER and PR negative, mainly will be aggressive form of breast cancer like MDMB231. Now, this cutoff score was set based on the publication from Dr. Jyoti's lab, where they had previously done this prediction score, and it was done with MCF7 cells itself. So the aggressive form, now our work was mainly done not with 231, it was done on MCF7. So we have to even maintain that homogeneity. Now, I hope I answered your question. Thank you, Divya. Can you comment on the specificity of circulating microRNAs for breast cancer subtypes versus other cancers or inflammatory conditions? Now, to address the specificity of circulating microRNAs is that you need to identify, are these originating from the breast tumor tissue? Like, you have to address the disease specificity of the circulating microRNAs. So that's an ongoing research that we are currently taken up, because our lab was the first one to identify the presence of miR-195 in luminal A breast cancer patients. So it was detected in the blood. But if you would look at the tumor tissue, miR-195 is a tumor suppressor microRNA, which is actually downregulated in expression. So there is a discrepancy if you try to correlate both. But I would like to say, like, ideally, if you can identify the presence of circulating microRNAs through EVs, that's a solution to it. So I hope I answered that. Akash, hello, ma'am, wanted to know who are the main victims of suffering breast cancer more in India. I didn't actually understand the question.
Is it related to age or any demographic?
Oh, okay.
I don't know.
That we'll have to look at the epidemiological data, like published by probably WHO or Globocan. But even US the breast cancer ranks number 1. So if you look at India, I think it's cervical cancer, and then tops breast cancer is second or third to next to lung cancer. So demographics definitely play a role. Age also plays a role, but now like cancer is becoming more common in young people as well. So I wouldn't be in a good position right now to give an answer to that. But thank you for that question. Thank you. Thank you, Naya. Thank you. What bioinformatic tool is efficient in analyzing the mRNA-miRNA interactions? There are plenty of data sets or the tools which you can try for this. Tar base, like a lot of things are coming up. And I would definitely say theraCUES would be the best people to contact for that. Like I would say Manjusha. So they were quite supportive in doing it. You have Cytoscape, so many other like tar base. So you could try all that. You'll be validating the MIR, so independent patient samples. Which type of normalization did you perform for your analysis? So for array, I think that was the nanostring array. So there was like a universal plate normalization done for the array. And other than that, we did not do any. So there was like a normalization with 10 MIRs. So the more details would be there in the publication as such. So I hope I don't see any other questions now.
Are there any miRNA targeting therapies currently in clinical trials showing promising results for metastatic breast cancer? There's a question by Akash.
The first part of the question, isn't it? Specific miRNA targeting therapies. I'm not sure if it has reached clinical trial, like could be in phase 1, but not currently going to phase 2 or 3.
Yeah, just I wanted to, can you hear me now?
Yes.
Yeah, this is Gopi Gopalkrishna. So,
okay.
See, I saw your interesting thing, ERPR positive converting into TNBC. Does it happen in the patients also, or is it?
It does, because right now, as I mentioned, like we have a biobank where we get the samples. So even in the same patient, when they come with remission, luminal A turns to HER2, or the next second tumor is triple negative. So it's an entirely different tumor. Even in the contralateral tumor, it happens like that. In the same patient, the left breast would be another phenotype and the right breast would come with another phenotype of tumors. So that's definitely possible. And that's what we showed, like even in oral. Now, oral doesn't have much hormonal combination. So what we could see was the presence or absence of the hyperplastic chemoresistant fractions. So the final conclusion would be to identify resistant cells. Whereas in breast cancer, you have a phenotype based on which you actually determine what treatment regimen should be applied to that particular patient. So there, this phenotype really plays a major role.
Okay.
I think I missed 2 questions. In addition, can you also comment on if you identified any specific microRNAs that regulate breast cancer stem cells directly? And could these be therapeutic targets? I think those are all ongoing research. So there are no like specific microRNA. There's a panel of microRNA. Now the problem with microRNA is that one microRNA can target multiple genes. So even if you use one, there will be another one to support that functionality. So if you withdraw one, there will be another like that. So there should be a unified study with microRNA and the mRNA, and need to correctly identify. Now, as I showed in the slide, the one with HER2. Now, HER2 receptor itself can trigger an upregulation of 3 or 4 microRNAs. So that can trigger so much of pathways downstream. And probably it's something we could look into in future. Thanks, Divya. Another Divya. What are the limitations and challenges in targeting microRNAs for breast cancer therapy? Now, that's the topic for a review. There's, as I just mentioned, like the limitation would be it's like a small quantity in tissue. So the extraction, the identification itself is a bit time consuming, and molecular biology work, so it has its own disadvantages and limitations. So getting proper samples and the sample size, you can't do validation with 50 samples. So we all often revert to current data sets like TCGA or Metabrick. So sample size is of concern. Then targeting microRNAs, you definitely need to have a specific microRNA which would prove like, okay, targeting this would work. So I hope I answered your question. I just want to have you looked into the role of circular RNA? No, because that's kind of latest. So microRNA itself is under the regulation of long non-coding RNA and circular RNA. So they are like parallel studies that could go from here on. So thank you so much for asking that. Your thoughts on maybe microRNA clustering? Yes, definitely. And now the therapeutic angle, like there is a group at LAM led by Dr. Roshin Dwyer. So they focus on miR-379 cluster. So that's like a tumor suppressor miR cluster, and they're trying to see if reintroducing those can actually suppress the oncogenic function of tumor. So it's still in vitro cell line study, so it has not reached the clinical trial. So thanks, Divya, for that question. Dr. Ramdas, what comments on use siRNA therapeutics? It's seemingly good at the moment, like very promising. Like when you look at silencing, it works well in vitro cell lines. So it's in a regulatory setup, it works fine, I would say. But we need to like prove whether the same functionality applies when you try to do it in a 3D model. So people are now working with 3-dimensional organoids and stuff to see. But I would say the final validation happens when you recreate these effects you see on a plate in a multicellular organism, where you will have to face immune interactions, the immune rejection. So whatever work we are currently doing is all in NODS-KIT, which is immune suppressed. So the bringing in the immune cell changes the scenario drastically. So thanks, Ramdas. Okay, I see Divya. Can therapeutic modulation of microRNAs reverse? Yeah, therapeutic modulation, we have not reached there as far as I'm concerned, but you can definitely do that in vitro and cell lines and tissue culture flasks. So definitely you can do. Role of microRNAs in tumor and tumor microenvironment. Obviously, the microRNAs from tumor tissue get secreted out as EVs. So they do modulate the tumor microenvironment as well as immune cells. Now, the profile of the EV microRNA is of major concern. Now, as there was a previous question, like how do you identify the tissue specificity? And that's actually a good angle to look at, like, can these microRNAs, where do they go, which stromal cells do they attach to, and what are the functionalities they do? So probably they can even function like a viral RNA, where there is like multiple mechanisms happening when a virus infects a cell. Since microRNA has multiple targets, how can we validate one specific microRNA majorly regulating a target? So I would never say there is only one microRNA regulating the target gene. Now, just from our array, we found that particular microRNA miR-29 to be quite upregulated in comparison to others. But there may be other bypass mechanisms of microRNAs which can trigger parallel signaling pathways taking up at the same time. So all that has to be done in a 3-dimensional model to make sure, is this the one that could be taken up for therapeutic angle? And I hope I answered all the questions. Thank you so much for the questions. Okay, Dr. Gopi, since tumor itself is heterogeneous, what is the effect of mice on tumor growth? Okay, does host environment determine the fate? Environment definitely determines the fate. Now, when you run or when you inject different fractions of cells to study the tumor growth, you always run a control as well. So in the control, if there is any influence of the host, it definitely gets reflected. So in comparison to host, if there is anything else happening, changes happening in the other fractions of your interest, you will have to nullify that. So that's why we do have tumors such as immune cold and immune succumbing like tumors. So they do react to immune cells. So whatever studies so far, as I suggested earlier, it's all been done in immune compromised. So the next phase of getting microRNAs therapy ready would be to establish that functionality in immune activated mice or not non-compromised mice, for instance.
Mm-hmm. Yeah.
So I hope I did answer the questions.
I think there was quite a bit of questions. Thanks for answering all of them. Yeah. Vijayalakshmi.
Yes. Thank you, Dr. Vinitha Richard. That was very exciting, and we've interacted with some of the other co-workers that you've had mentioned in your slide.
Okay.
So yeah, we've been working on miRNA as a detection. I think I'll be just telling the audience what are the different ways you can detect and find out about the miRNAs that are there in your samples. So exosomes, for example, have a sizable miRNA population, and that is being used as therapeutic in a therapeutic angle. And most of it you have already mentioned. I would just reiterate that these are post-transcriptionally, they modify the mRNAs, leading to degradation of mRNA as well as translational inhibition. So their roles are being developed, I mean, in development or differentiation or disease, or even in circadian rhythm, many in many cell biology. It has been like, every day you find out new roles for these miRNAs. And in fact, in 2024, the Nobel Prize for Physiology of Medicine was jointly awarded to Dr. Victor Ambrose and Gary Ruvkin for the discovery of microRNAs in the year 2000, if I'm not wrong, and for its role in post-transcriptional gene regulation. So these are the molecules which are playing a pivotal role. As you already mentioned, that one miRNA has a cascading effect. And how do you detect these? So there are multiple methods. qRT-PCR is one of them, sensitive, of course, quantitative, and it requires reverse transcription, but still this method is widely used. Microarray, you've already mentioned that. Then next generation sequencing is what most comprehensive people are using more often than not. And it can detect novel miRNAs. So this is another method. Encounter, as Dr. Richard mentioned, can detect 800 miRNAs of human, and even mouse you can do this. Highly sensitive and very reproducible. So we would encourage people to, utilize this technology which is there in India. Coming to you from theraCUES, we are the first ones to provide services for Encounter and GeoMx. GeoMx, of course, we've already spoken in our previous webinars about it, but Encounter is a very promising technology to detect miRNAs. So I will just briefly about the technology, how it works. miRNA, as you know, is only 19 to 22 nucleotide long, and so you add a mir tag, and there's a bridge oligo which extends the whole miRNA molecule. And here you use only two probes, a reporter and a capture probe, to count these. Basically, it's an encounter, so it counts. There's no sequencing involved. There are no enzymes involved. There's no cDNA synthesis. There's no adapter, or all those things are not involved. So it's a very simple technology and very powerful at the same time. So this complex that is generated, so this DNA RNA chimera, the miRNA and the tag, along with the capture and the reporter probe, together forms a complex which is detected by the encounter machine. And the assay takes barely 2 days, and so it's very fast, and hundreds of samples can be, and in fact you can use RNA from FFP samples, and on any kind of samples like exosomes. We've worked on those samples in the past. So Dr. Richard already mentioned that these are used as biomarkers. Therapeutic angle is still being explored, and developmental biology, of course, it plays a very key role in gene regulation and drug response, so predict the outcomes of a drug therapy. So all these are the areas where many scientists are working on worldwide. And theraCUES has been working with miRNAs for long now. It's been 7 years in operation, and we've had a lot of publications. You can go to our website and check what are the work we have done on miRNAs, both in from NGS point of view and from encounter. So it's yeah, that's what I wanted to mention. So all of you can visit our website and see what services we provide. DSP is one of them, which is so targeted gene expression through encounter. We do cell line authentication because cells have to be in proper state before you even start your experiment. Then we take independent bioinformatic projects, of course, and custom solutions and extraction services for RNA, because RNA extraction is not easy from all samples. So I would encourage all of you to visit our website and contact us for any of the services. miRNA being the molecule for the month, definitely you can explore that through Encounter and NGS platforms that we offer. If you have any questions for Dr. Vinitha, I would ask you to. Yeah.
There is one more question there, Dr. Vinitha. Sorry, you had to answer.
Yeah, 1 second. Yeah,
Rajinder
Kaur, is that correct?
Yeah.
I have microRNA and proteomic data of 2 groups. Okay. I want to analyze the effect of miRNA regulation of protein expression. So you just have to predict the target, like the mRNAs, these microRNA can bind, and obviously correlate your target with the protein. You don't have to go and directly connect with miRNA or protein. So just for protein database, you have string and David. So I don't know if there is anything existing where you can do both together. But for me, logically, it looks like your protein and the mRNA could be related. So you can predict the mRNA interaction, and then you can bring it to the protein level. That's it. I'm not sure if anybody from theraCUES would be able to answer that part, like if there is a specific tool or software that can be directly connecting miRNA and protein.
Yeah, I think what you said is correct. Maybe mRNA, we should first look at it and then take it to the protein. So that would be right way to take it forward, because they don't directly target protein, anything at this point, only mRNA at this point. So.
Yeah.
Is this another question which is there? Is the prognosis during tumor enlargement identified using any specific tools that are not available in India?
Prognosis during tumor enlargement.
Identified using any specific tools that are not available in India?
So prognosis is the outcome. So basically, I don't know, that's like a risk assessment. I didn't really get the question. tumor enlargement. Basically, you do mammogram, isn't it? So other than that, I don't know. I'm not sure what the question meant actually.
Yeah, maybe the progress, that's what they are looking at, I think. So.
Okay, so the outcome would be the tumor size or the involvement of lymph node actually indicates poor prognosis. And if you look at more, microRNAs, you could definitely find microRNAs which are like a signature for metastatic patients or like people with recurrence. So if you have a panel which can identify those, that could be used as a tool to detect it. I don't know, like tumor enlargement, like people surgically it gets resected, so or maybe recurrence. I really didn't get the point there. Now, as a tool, I have really no clue. If it's
Yeah,
diagnostic, we have this MammaPrint blueprint and what is it, oncotyping. So those are all mRNA-based assays.
yeah. I think probably she was looking at any device kind of thing are they looking at. So that's what probably. Yeah.
I'm not aware of. Sorry for that.
Only the imaging technique is the only thing which presently.
Mammogram, isn't it, or ultrasound probably.
Yeah.
But even like mammogram is not used on women below the age of 40. So even for young people who have cancer, so obviously they do detection, self-detection or self-check, breast check. That's what mainly they say. So that's the simplest thing that people can do. And that's what they even use here. Like people who have the breast check or frequently, like when you have some doubts, approach your GP or things like that.
Sure. Yeah. Mm. I think we are coming to the end of the webinar. I think Dr. Vinitha, fantastic talk. We really enjoyed, all of us. If you look at the number of questions, you can see how many of the questions have come. So it was quite informative. And then, though we did the project, I didn't get into the project details clearly. So I think Manju was involved quite a bit, and then Vijayalakshmi was in the beginning. She was working together. So thanks a lot for working with us and then being a great speaker. And then I hope you have. I'm sure there are a lot of people have got inspired by the kind of work you've done. I know there is a lot of efforts are there, but the way you told is very highly simplistic thing. I can see the efforts which has gone behind. So that is, it's not so easy to do this kind of experiments. I can see it. So fantastic. And then as future, let's look at it. How do we collaborate more, and then how we can work together with multiple platforms what we have at this point. So we thank everyone who are at one point around 78, 80 people were there. Now around 50 people are still around. So thanks for joining this webinar. It's fantastic, and then we'll be doing more these kind of educational stuff. Last month we did miRNA, and then next month we are going to have an anniversary talk, and then we will be doing more and more educational stuff. So thanks, Dr. Vinitha. If you visit Bangalore, please do come and yes.
Thanks to all. Thanks to theraCUES as well for this opportunity. Thanks to all for your patience.
Yeah.
Thank you.
Thank you. Thank you. Thank you, everybody, for participating.
Thanks. Bye.
Thank you.